Hello, everyone. Today I'll talk about Neolithic mortuary practices in cave burials. I'll briefly go through the aim and the outcomes of the research and present a short background of Neolithic cave research in Britain. I will mostly focus on the methodology that I used, its importance, and present examples of preliminary findings from some of the caves I examined. The aim of the research is to contribute to the understanding of mortuary practices during the Neolithic, from about 4000 BC to 2000 BC, by examining the macroscopic and microscopic characteristics of human remains deposited within caves in southwest Britain. Taphonomy is the study of the agents and processes that influence the body from the moment of death until remains are discovered. More specifically, macroscopic taphonomic analysis provides information on the degree and duration of exposure of the remains, the nature of manipulation and/or disturbance of the bones, and the agents of these modifications impacted on the bone. Microscopic taphonomic analysis, on the other hand, indicates variations in the bioerosion of bones from burial contexts and provides insights into the early postmortem processes occurring in burial. By combining traditional and novel taphonomic osteological analysis, this research will examine pre- and post-depositional treatment of deceased at or near the time of death and after death, and clarify the means by which bones became disarticulated. This is just an example of the different agents that can affect the body in burial. Both human and natural agents are presented here, which shows how complex burial processes can be and the number of different agents that can impact the bone until we get to examine it. Caves are one of the many contexts where Neolithic human remains have been discovered. A corpus of information, including 160 directly dated individuals to the Neolithic across Britain, and additional cave reports, provided an impetus for research on Neolithic cave burials. These researchers have privileged osteological and theoretical analysis over ritual activity and have demonstrated a bias towards regional analysis. For example, monumental versus non-monumental burial debates have been centered on Northern England. Isotopic analyses have attempted to characterize the dietary preferences of people in Wales, Somerset, and Devon. However, the scattered and isolated nature of the evidence means that more work is needed to fully compare the life weights of individuals buried in monuments and caves. In particular, there is a rich resource of unexamined human remains available in Wales that forms the focus of this research. There is also an abundance of human remains available in the Mendips, as one of the largest concentrations of caves in southwest England has been discovered there. Full taphonomic evaluation of sites outside of Wales is beyond the scope of this research. However, a targeted histological analysis of already radiocarbon dated remains that benefit from existing isotope studies offers a good opportunity for a comparative study. Macroscopic analysis has been undertaken on 20 sites in Wales, with microscopic analysis of 7 of these sites and 1 comparative cave from North Somerset being conducted. This will lay the groundwork for future research by identifying any patterns suggesting a more widespread funerary process or variations suggesting regional or even localized traditions. Furthermore, cave archaeology is riddled with challenges surrounding chronology and depositional history, and these have severely limited the development of studies on prehistoric cave use. Therefore, the substantial corpus of Neolithic human remains has been subject to limited holistic analysis, and research is inhibited by poor chronological resolution. Caves do not have typical depositional patterns, with material reworked by cave hydrology and consequential movement of interments, disturbances caused by human occupation and scavengers, and excavations from non-archaeologists and cavers. As a result, stratigraphy is not a reliable index of chronology, and radiocarbon dating is crucial in understanding the history of use of these sites. Accurate dating of human remains supports research into cave archaeology and enhances our understanding of the occupation and uses of caves throughout time. In order to achieve that, 10 skeletal remains from 7 sites in Wales were submitted for further radiocarbon dating. Whilst caves constituted one of the burial contexts where people interred or disposed their dead, tombs have been the most tangible evidence of Neolithic people in their customs. This evidence has helped us portray portions of their living conditions in prehistory, get a better understanding of the attitudes towards death, and compare the distinct burial contexts. The multiple burial trends, with a predominance of disarticulated, fragmentary, and incomplete individuals, have amplified suggestions of general circulation, movements, and arrangement of human skeletal material in Neolithic Britain. This uneven representation of human remains has created imprecise understandings of the way people handle their dead. Thus, the reconstruction of mortuary practices is truly important, as it can help us get a better understanding not only about the treatment of the deceased, but also about the living. And what is truly interesting and evident throughout the Neolithic period is the diversity in which the dead are treated. So during the early Neolithic, we find bodies that are left out to naturally deflesh. It's called excarnation. That's a practice of removing flesh and organs of the dead before burial, leaving only the bones. We have remains that are moved and placed in collective tombs or caves and rock shelters as part of secondary burials. Bodies that are moved to make room for more interments in reused tombs during the Middle Neolithic, and single burials and cremations that alter the traditions in the late Neolithic. So whilst we have diverse evidence in different burial contexts, surface modifications of human bones can help us understand the degree and the duration of exposure of these remains, the nature of manipulation and or disturbance of the bones, and the agents of these modifications that have affected the bone in different burial contexts. Next, we have some preliminary results from the microscopic taphonomic analysis of 5 sites. I'm not going to go through site details as we don't have much time, but rather focus on the taphonomic trajectories of the elements. Remains of about 7 individuals, including 5 adults and 2 juveniles, were discovered in a crevice in Ifton Quarry in 1908, primarily consisting of human crania with a few accompanying postcranial remains. 4 radiocarbon dates have confirmed middle to late Neolithic activity in the site, and no other finds were present. Evidence of human-inflicted trauma appears on most of the skulls. That includes blunt force trauma, cut marks, a healed fracture, and possible scraping, defleshing marks. This is also supported by the presence of dry fractures on the long bones that resulted from later disturbance, possibly anthropogenic. Remains were recovered from a narrow area underneath a projecting shelf of limestone near the summit of a steep slope, meaning access to the rock shelter was limited and consecutive visits could not have been achieved. Evidence therefore suggests that the site was used for final deposition after remains underwent a series of processing. It therefore appears that we have a multi-stage burial of individuals of different ages. Disarticulated human remains of about 4 individuals, including 3 adults and 1 juvenile, were discovered in a small, highly disturbed area in Novo Kolomenti during excavations that started in 1975. Remains were mainly discovered by cavers and the excavator. The human remains were mingled with animal bone and 1 patinated broken Neolithic flint arrowhead. Currently, 2 radiocarbon dates are available from the site, 1 Middle Neolithic and 1 Early Bronze Age. Extensive evidence of animal gnawing and both fresh and dry fractures confirmed the high level of disturbance in the site. The state of disarticulation and the large quantity of fractured bones has led previous researchers believe that this was part of a funerary rite. So the number of human and animal remains seem disproportionately large for such a small site, with the majority of animal remains being similarly maltreated, fragmented with cut marks based on the excavation report. New radiocarbon dating evidence from 2 elements will further clarify patterns of mortuary deposition in the site and help us understand how it was used for depositions. Spiritual was discovered in 1985 at a steep cliff in Swansea, and human remains were unearthed at the entrance of the cave. According to the excavation report, part of the skeleton was discovered near the surface at the mouth of the cave, eroded and disturbed by burrowing animals. So remains were indeed very eroded, very crumbly, almost dissolving as soon as you pick them up, with a lot of root etching. Now, this is a very interesting case of burial. Whilst with this appears to be one individual, remains were found in the correct anatomical order. On the basis of morphology, both female and male elements are represented in the site. So we have a right female pelvis, a left male pelvis, and then one femur is more robust than the other. Loose teeth of an older male and one deciduous tooth from a juvenile are also present. Therefore, we have about 3 individuals present in the site, so 2 adults and 1 juvenile, and only 1 early Neolithic radiocarbon date available. 2 upcoming radiocarbon dates, however, will further clarify the period of use of the site. It has also been suggested that this individual is reconstituted, so people visited the site over a period of time and deposited the different elements of the body to make a whole new one. There is evidence of this happening during the Neolithic in chamber long cairns in southeast of Wales, in the Black Mountains. And what I find really interesting is that the preservation of the remains is the same. So all the long bones, cranial fragments, as well as the teeth have the same preservation, which means that they were possibly all deposited around the same period of time. And whether this was part of a ritual or not, they were all preserved in the same manner. It will be very interesting to get the results from radiocarbon dating and examine the microstructure of these elements to test this theory. The famous Gop cave is encapsulated underneath a cairn. It was first excavated in 1886 with finds dating from the Paleolithic to the Neolithic period and more than 14 individuals originally discovered. The remains have since been lost. Therefore, we have an MNI of about 5 individuals, so 3 to 4 adults and 1 juvenile based on these skeletal remains currently being stored at the National Museum of Wales, and 3 radiocarbon dates ranging from the Middle to the Late Neolithic. A few more elements, including 2 craniums, are stored at Manchester Museum. Same as in the case of Little Hoyle Cave, which I'll show you and talk about in a minute, taphonomic indicators varied a lot on the elements recovered from the site. So we have cases of concretion, different degrees of black staining, some evidence of weathering and fractures. We see, for instance, cases of cave concretion on some of these elements, not all, which covers most of the surface of the bone. Initial reports mention them all being deposited in one chamber, with a few elements discovered at the mouth of the cave. Evidence of distinct taphonomy on the surface of the bones can be explained with the periodic use of the cave. So if secondary burials did occur, remains were exposed to different agents before being deposited in the cave. This means that this is a repeated pattern of deposition in the site. There is also not enough evidence of gnawing on the bones to suggest disturbances from burrowing animals. However, few elements show evidence of weathering, quite unusual for cave burials, which suggest potential subburial exposure before final deposition. Last, we have Little Hoyle Cave. About 10 to 11 individuals, adults and young adults, were discovered in this small cave, which was first excavated in the 1870s. 4 early to middle Neolithic dates, with one forthcoming radiocarbon date, suggest a lengthy period of use of the cave. This site had the largest quantity of mandible fragments recovered from all sites examined in this research. The human remains were discovered in an infilled shaft, a chimney that connected the cave to the entrance, and were not properly recorded at the time. The number of individuals suggests successful episodes of interments, along with the animal bones and a variety of artifacts, pottery, and metalwork discovered, dating from the Upper Paleolithic to the Roman and British period. The surface taphonomy of this site varies a lot too, so very distinct evidence of black staining, either manganese or mold staining, can potentially suggest exposure to different sediment types or environments. Furthermore, the extensive amount of mandibles recovered and the low number of cranial fragments and long bones poses serious questions about distinct treatment of cranial versus postcranial remains. Mandibles are one of the first elements that disarticulates from the body, And since only a few cranial fragments, including a reconstructed cranium, were discovered from the site, two trajectories seem possible. Either remains were circulated, so craniums and other postcranial remains were removed from the cave as part of a mortuary practice to a new context, leaving the mandibles and other smaller elements back in the cave, or preserved elements were brought disarticulated in the cave from another context for secondary burial. So now that we have all these results, what's next? As I mentioned, microscopic taphonomic analysis provides insights into early postmortem processes and reveals the rate and nature of soft tissue decay. It can show the different degrees of bacterial attack on the bone collagen, and the microstructure of the bone is like a descriptive map that sustains information throughout the years. This analysis is key in understanding the distinct types of funerary treatments, since the degree of bioerosion, bacterial attack on bone, can occur either through gut bacteria released after death whilst the individual is buried, or from possible exposure to different soils after disarticulation. So good preservation, this will look like this, of the microstructure indicates possible disturbance that did not allow the bacteria to attack the bone. And bad preservation is evidence for complete primary inhumations. No matter the variation of preservation, the Oxford Histological Index grading from 0 to 5 allows us to ascribe a certain degree of preservation and interpret these results. So, for example, if the soil-stained surface of a bone was facing up and was not in contact with the ground, this would be a clear indication that the remains had been disturbed. However, due to the level of disarticulation and disturbance of the bones, we cannot accurately confirm the treatment that the remains underwent. However, by analyzing the microstructure, we can get an understanding of the processes that affected the bone. If the preservation of the microstructure is good, then it suggests that disturbances, secondary burial, or possible circulation of bones occurred. However, if the preservation of the microstructure is bad, then it's a very strong argument for primary burial. Another good example is the case of cut marks or trauma on the bone. If the preservation of the microstructure of a particular sample with evidence of scraping is good, then it's a very strong argument for defleshing. Hence, according to these results, we would be able to understand whether the cut mark or scraping marks were the result of defleshing or a perimortem trauma. So to summarize, by combining all these analyses, this research will establish the early postmortem treatment of humans and differentiate practices of inhumation, exhumation, and excarnation, will differentiate treatment between the individuals, building on the microscopic analysis that has already been conducted, and reveal any regional variations in early postmortem treatment of the disarticulated remains from Wales versus North Somerset. So the results and the combination of macro and microscopic analysis can disentangle issues of pre- and post-depositional treatment, an aspect that can be confidently answered otherwise. The results of this study will not only redress issues of mortuary treatment in the UK, but will finally create a more holistic view of how prehistoric societies function and potentially explore patterns in social structures and treatments of the body. This will not only alter perceptions from a scientific aspect, but will also attach importance to current theoretical studies. Furthermore, the combination of data and theory will redress the imbalance of previous research and build on the existing extensive stable isotope datasets. Thank you very much.